So I think we're going to make a start, everyone. So I have the pleasure today of introducing Edith Hammer, who is is currently a senior lecturer and AP at Lund University in microbial ecology. She's a PI at the Biodiversity and Ecosystem Services and the Changing Climate Lab. And I'm very excited to introduce this talk because I've been following Edith for quite a number of years and seen some of this work beforehand elsewhere. And it's absolutely fascinating and had everybody on the edge of their seats. So I'm really excited to be able to introduce her today to talk to everybody. Edith has a special interest in the effects of microscale soil structure on nutrient cycling and carbon dynamics and on the mycorrhizal symbiosis. And her group has been using the microfluidic chip systems to simulate the environmental conditions and symbiotic interactions found in soils, allowing for video capture of microbial decision-making and behaviors. Oh, I don't know if the mic's on, I've got a comment from online. Just before we jump into Edith's talk, I just wanted to give a little plug to some of the other talks that we have upcoming in the Nature Seminar series. So on Tuesday, we have Anna Carolina Antunes coming to talk about Amazon flux, where she'll be talking about her work disentangling biodiversity ecosystem function relationships through energy flux perspectives. And on Friday next week, we'll have Jerome Lewis, who will be talking about building collaborations with indigenous and local communities using extreme citizen science. So please do come along to those too. And I would love to now bring up Edith to give her a fantastic talk. Thank you.
Thank you. Thanks. It's a pleasure to be here. It's my first time in Oxford, and I'm really blown away by the city. It's amazing. And I also, like, we were just in the park talking about how important it is to have nature close by. And I wonder, like, when was last time you have been digging into the soil with your hands, maybe even? Like, when have you got your hand dirty? Is that something you, yeah? Yeah, that's great. Because, I mean, like, then you probably agree with me and I think it's the most fascinating ecosystem we have on Earth. It's, you probably have heard that there's more organisms on a spoon of soil than there's humans on Earth, and it's the species densest habitat on Earth. And there's also more than twice the amount of carbon in the soil compared to all the living biomass and the atmosphere together. So there's a lot of superlatives. And what makes this ecosystem so special? Like, how do we get this huge accumulation of carbon there, for example? If we compare a water ecosystem to a soil ecosystem, there is also free carbon in water ecosystems, but so much less. It's like 3 orders of magnitudes less carbon compounds in water ecosystems than there is in soil. But like here, nanomolar and micromolar. But in addition to the huge differences in the carbon accumulation, we also have way more organisms in the soil that are super hungry. They want to eat that carbon. There are hungry organisms in the water system as well, but they are much better at taking what is there. And if we, but the soil microbes are seriously hungry. If we add some very easily available carbon from the outside to the system, it goes like, and it's being respired within a few hours only. It takes much longer if it's diluted in the water body. And to my mind, the reason for this big difference and for this huge accumulation of carbon and of species as well is the microstructure that we have in the soil. This is flight generated through a soil aggregate constructed by John Crawford based on a micro-CT measurement of a soil aggregate. And there you can imagine what your life as a microbe might be if you have to find your food in this labyrinth and also try to not become food yourself at the same time. And I have been interested in physical soil carbon stabilization for a long time. So the part of how can things hide in the soil ecosystem? Can organic matter be spatially hidden from decomposers, and what is spatial availability for microbes to start with. So it's a little bit like this Pac-Man game from the 80s. But very often when we study soil ecosystems, that's what we do with them. We completely destroy the structure, and that has a lot of good reasons. I mean, like if we want to have a more homogeneous data set, and for a lot of questions, it's maybe not so important, but we miss a very important, very, very intrinsic characteristic of a soil if we destroy the structure each time we do experiments. And that's why we have been developing these soil chips. Those are transparent micro labyrinths that we can integrate into the soil. We can either put them right down into the soil or take soil into the lab and then have this like a cyborg system where we have a technical, easily microscopical window to the soil. That's the way we construct them. We start with designing a spatial design for a certain research question, and then draw this in AutoCAD, transfer this to a photomask, and produce kind of a stamp that we pour a polymer over that is breathable. It's good culturing conditions and so on, and takes all these microstructures down to nanometer scale can this material take. And then we can study things under the micro. You're right, this was us in the clean room when we produced the masters. And then we can study ecosystems with soil ecosystems with these, either by having cultural strains and small model communities, or having the whole soil put as an inoculum. We have been working with them a lot as static systems, really as labyrinths for quite some years, but we're also getting into flows recently. And we think that these micromodels represent at least some parts of the soil microhabitats quite well, because we have these very small structures that represent the world that the microbes experience much better than if we have them on a plate or in a tube. The world at the microscale is quite unintensive to what we know from our everyday experience. We have very much higher impact of surface tension and capillary forces. Flows that we experience usually is turbulent, but flows that microbes experience is laminar. And for them, going through water is like for us maybe going through honey. And a water bubble, oops, I wanted to run this video. Can be completely impossible for bacteria to go past, even though if it's only a small water bubble, a bacterium will never be able to pierce through the meniscus of the water surface, even though there's a fungus that very happily just grows through. And we think that these microsystems can be quite a handy tool for soil ecology, because we can simulate a lot of factors that otherwise have been difficult to do, simulating physical heterogeneity, but even chemical. We can get a lot of different microbes in there to study interactions. We can also get roots in there to study plant microbe interactions. And there's this approach of culturing the unculturable, where we have a system that allows for flow of metabolites, but having still spatial separation so that things don't grow dominant too easily, so that you can culture up to like 50% of the soil microbes suddenly, which otherwise is more like 2% or so. But back to the first question we tried to answer: what is spatial availability for microorganisms? We started with designing a labyrinth chip, which was kind of 5 experiments in one, where we wanted to test different spatial patterns and how fungi react to this. So we had angles, widths, pore spaces that opened up, and so on. And we have been inoculating them with 1 to a handful of different fungal species. And I guess a few of you are mycologists. So I'm giving you a little bit of help of remembering what species we have. We had quite some different foraging strategies in them. We have these Coprinellus that is a little bit like a marathon runner, very, very many hyphae, very fast growing, far growing. And then we had this little funny guy, Psilocybe, that was growing very giggly and didn't really seem to have a plan often. Then we had our zombie fungus that was going with a super dense front and like plowing down even parts of our chip and destroying them really, really strong. And then we had this one, the snaky one that liked to grow and get stuck in corners and do these snake structures once in a while. And they have quite different strategies to explore space. Here, our zombie goes with all fours and almost grows like the water flows at the scale, while others are exploring much sparser. Also, it's good to know that there's no reward in there. They're just growing in air. So they're just really exploring where they maybe can find something here. And if they are offered with a little bit more space after a long time of constriction, we wanted to see, do they take that space? Do they branch in these spaces? And we found a couple of species that did, and a couple of species that don't, like pretty consistently at that scale. This is like 150 microns here. And then we thought there was some previous work saying that hyphae want to have an obstacle to induce branching. Does that happen then? If there's a pore and there's a little sand grain to come, then they explore the space if they are forced to take a decision and then branch? It was a bit the same, like those species that always branch, they branch also happily when they hit an obstacle. Those that usually don't branch, they rarely branch, even though there's an obstacle. And we wanted to know what are difficult structures for fungi, like where in the soil pore space could be areas that are very difficult for them to access. And we have been looking, yeah, it could be that fungi like to go radial and straight. Maybe they don't like to go through sharp angles. So have been setting up channels of different angles. And that was true for most species. Most species don't like to continue in channels that have acute angles, except for the snaky one. It loved it. And it grew much, much further in the very windy roads than the other even. Not even didn't care, but it preferred them. So it was funny to see. And this is how it looks live in a time lapse. So it's totally okay with these 90 degrees, which are in the direction of the growth. But, but already if you just turn the 90 degrees that they sometimes are 90 degrees towards their growth direction, and it's very difficult for a lot of species already. So it really gets stuck there and kind of clogs this whole channel until in the very last part of that weekend when we took that time lapse, it found a way to continue. And we have also been putting avascular mycorrhizal fungi into these chips, which was quite complicated because of all the plant part that you also need to have, but we managed in the end. And they have a very distinct way of space exploration. And I mean, they have their carbon supply from the plant, so they are not carbon limited in systems. And we thought that they maybe are even more prone to go and scout around because they maybe have the opportunity better to go and look into spaces that are less attractive for saprotrophs that always need to find energy resources as well. We found that also these angly roads were not very preferred by the avascular mycorrhizal fungi. But it was very funny because in these hyphae, you can really see the decision, like it tested it for a couple of micrometers and then saw, no, there's nothing for me to get here and it's getting uncomfortable. Then it takes all its movable biomass, all the cytoplasm back and translocates it somewhere else. So here we have an intact hypha and here we have a retracted part. And it does that very, very frequently. Here's a bit more complex part, and you can see the cytoplasm streaming, but you can also at the same time Now let me see if I get this run. We have here a detour that is cut off, wasn't deemed necessarily here as a, here is a, no, I do like this. Here's a corner, there was nothing to get. So those parts are abandoned again. Also, if you remember the other fungi exploring or not exploring open spaces, and that were pretty much programmed by their species identity of what they wanted to do, that's very different. Like there's a strong responsiveness in this avascular mycorrhizal fungi that reacts very strongly to obstacles and was like branching almost every time when there was an obstacle coming in its way. And the funny thing, I don't know if anybody has ever worked here with cultures of avascular mycorrhiza, maybe you? Because I was super amazed to see that they're doing this. They are producing spores in our chips. And usually they are these beautiful balloon round balls when you sieve the soil that you can use for species identification as well. But they form them in this labyrinth space as well. And they take the shapes of the space that they have, making me realize, like in my master thesis long time ago, I have been counting spores for estimating inoculums. We probably rarely get them out, and this is also probably the reason for that people see that avascular mycorrhiza really change soil properties, for example, the water holding capacity, because they change the pore space distribution quite a lot then. Then we wanted to go the next step further and not only look where can microbes get and go, but also what is happening with organic matter in different spaces. So for the next series of experiments, we have been adding fluorescent organic compounds into the system to see where and how fast and to what extent organic matter is being used and degraded. We have been doing this in a neat model system using one fluorescent bacterium, one fluorescent fungus, and then a fluorescent substrate as well. We started with a chip that had some more variations of angled roads, and our hypothesis was an easy path for microorganisms is something that they like to find their food. But if the road gets more windy, they get a little bit angry. And if it gets even worse, they get really angry and think this is too much effort. So our hypothesis was the straighter and easier the access to food, the more easily and the stronger it will be used. And we hypothesized that would be the case for fungi because we just found these things in other experiments. We were not so sure how we would find this with the bacteria because we thought they can go everywhere. There's a water connection, so they can really go everywhere. But we also strongly found less biomass, less bacteria, and also less organic matter degraded in systems where we only had bacteria. And we think it's a little bit because they're like vacuum cleaner robots. They go back and forth, and if they are in a windy road, they probably use more energy and resources to go into the wrong direction to exploit things, probably. So we have a decreasing biomass with increasing spatial complexity, and also a decreasing substrate usage, organic matter degradation, at least when fungi are there as well. But then a soil aggregate is rarely having super long windy roads only, but it's more like a labyrinth. And the next experiment we did was designing a space-filling fractal landscape where we have more or less connectivity of our paths. So we were using the Hilbert curve. In simple versions of the Hilbert curve, we have roads that are connected like New York City, maybe, squares going everywhere. And then we have something that's maybe more like Old Oxford, where there's a lot of dead-end streets and small alleys that are not connected. And again, our hypothesis would be that in a connected system, we have a higher availability, both spatial access and energy input to get to these resources than in a very highly intricate labyrinth system. And that was straightforward for the fungi. The more dead-end streets we have in the system, the more difficult it is for a fungus to go through. They do after a while, but it takes them quite a long time. So the system is being explored at a much slower pace. And here's a little time lapse again of fungi entering. And then we found something that we were not expecting. I was quite amazed. I'm not a person having worked super much with bacteria. And then suddenly we got these swarm pack hunting systems here in the system. And then also, especially in the more intricate labyrinths. And that was already hinting at something that we did not expect to start with. Because if we look at the bacterial biomass, it increases in the more complicated labyrinths, in the more dead-end labyrinths. And the organic matter usage increases very strongly there. That was a surprise to me, being not a bacterial physiologist, but it probably has to do with the onset of quorum sensing and enzymes not diffusing so easily, so that the organic matter degradation can be done in a much more efficient way, probably. So there's more work to do in this story. We have been looking into the higher spatial details because we have these labyrinths as a whole. We have a higher degradation activity when there's a complicated labyrinth, but how is it if we look into the very deep parts of this labyrinth, depending on how far a space here is from the entry? And then we saw that biomass, fungal biomass steeply declines, but bacterial biomass until the very deep parts first increases, but then decreases, and the same with organic matter degradation. So we have a strong enhancement in the complicated labyrinths, but not deep inside them. So there, the hypothesis of organic matter maybe being physically occluded again can hold. Next things we're doing, but that's all very recent, is to look at resources which are not spatially homogeneous, but patchily located. So we have started to build little nutrient patches that we want to play with. And also, instead of only looking at fluorescent reporters and so on, we also look at more detailed chemical changes especially with Raman spectroscopy, which we can do through the chips inside at the micrometer scale as well. But we have been doing quite some work now with real soil chips, where we use real soil as an inoculum. And we either can take this in and inoculate or incubate them in soil for weeks or months, depending on what we want to do, or we take soil into the lab, and then we can also do time lapses on the microbial community that comes into those chips. You see a GIF of a bit more than a month of experiment, where there was also, it was an air-filled system to start with. After a while, fungi come in and they drag water with them, and then the whole other microbial community follows. And you have a very high turnover of the hyphae as well. And working with soil, we realized, yeah, we have this Pac-Man allegory, but it's probably more like this. Does anybody of you know that board game? Yeah. For you that don't, it's every time it's your turn, you take this one of the cards and you push a row of this labyrinth, so it changes each time. So there are new paths emerging, and also new dead ends starting. And you can annoy your play partners by putting things into their way. And that is probably what's happening all the time in the soil pore space as well. Here is a shot of a chip that was drying a little bit. We had it out in the air for a while, and then water is being dragged out from the chip. And at this scale, it's really like a tsunami. And this is only a few nanoliters of water moving. But if you are in these small pores, this is having a really big effect. And we have been trying to quantify this a little bit as well by particle tracking. And I can speed this up a little bit. And I just told Chet that I also studied a bit of geography a long time ago, and I love spatial things. And isn't this a beautiful flood estuary opening up at the micrometer scale? There we have areas that are super high velocities, and if you are a microbe there, you better hold on. If you are just 50 microns away from there, probably nothing happens for quite a long time, but maybe you also don't get so much resources to your spot. So there's always this trade-off. But it's a very strongly heterogeneous landscape that the microbes are facing. And I also have been realizing more and more that decomposers are not only deconstructing things, but they're also constructing workers. They really shape the environment, glue things together, build things, build their world. And they are very important for the structure that we have in the soil, that are their world, but they have been contributing, creating it a lot as well. Right, not only by gluing, but also by pushing new paths. This was our zombie fungus that has been really destroying our chips here on each of those corners where it should turn. It didn't want to. I was asked by Jeppe to briefly talk a little bit also about synchrotron work. He's not here, unfortunately, but I now talk about this anyway. We have been looking at these gluing activities of fungi as well at the hyphal scale again. So we have been taking different fungi with different minerals and growing them this time on plates, not in chips, on optical windows like this, even with avascular mycorrhizal fungi, to then study them under the Dixon beamline at some different synchrotrons, looking at the chemistry of the exudates that emerge when different fungi get in contact with different minerals, and how the interactions between the cell and the mineral then is. So we can see that fungal exudates touch easily a mineral that they come across, and they even like float and flip across those exudates and start to cover the surfaces also on the other side of the fungi. And in some of the cases, some ectomycorrhizal fungi even induce reduction of the minerals. So we have an iron 3 to iron 2 reduction, which probably is super important for their way of accessing organic matter via radical producing. So that there is this gluing effect, but there can also be more chemical alterations induced quite strongly then by a single hypha. And we also have been recently looking at how a bit larger scale aggregates are built up by labeling batches of completely deaggregated soil with rare earth elements. And so we could, for example, label a batch with straw with one rare earth element and a batch with maize, very different carbon-nitrogen ratios. Which are benefiting more the fungal decomposition channel or more the bacterial decomposition channel, and see then when we mix them how newly formed aggregates are being constructed. And we see, for example, large areas of the maize pre-batches still coherent, while the straw batch has been intermingling more and being distributed to much smaller chunks in a newly formed aggregate. So the organic matter, but also the microbes are playing a very important role in creating the structure of the environment they live in. Something that I think is important, an important advantage of using those microchips in soil ecology experiment is that we can combine the very high controllability that we can do in lab studies with relevant ecosystem complexity. And we have, for example, been studying ecotoxicology questions by looking at how microbes react to nanoplastics with soil inocula. Where we can look at whole microbial communities and how they're affected by a pollutant, in this case, plastics. And we could see that bacteria, for example, grow much worse when they are swimming in a soup of plastics. And the same was happening with fungi, but only for a while. We realized that some fungal hyphae, the first coming into an area that is polluted, are actually attracting those plastic particles, and they are getting stuck on the hyphal threads. So the green here is the plastic, the red is the fungus, and the first hyphae have been cleaning up the whole environment there. That was quite amazing to see. We have been testing this under very many different conditions and also with other species and with soil inocula, and that was quite consistent. It was funny, the Swedish media took this up as the fungal vacuum cleaners that are our hope now for solving the plastic problem. I don't know if I would go so far, but it was very interesting to see that this was happening, that the plastic particles are adhering so strongly. We also have a couple of exciting master students working with different ecosystems. One with biocross from Greenland, where we have with climate change more time of the year where there is no snow cover, and then we get much stronger freeze-thaw cycles in these ecosystems. So we have been simulating what happens if we mess around with freeze-thaw cycles of these biocrusts. And the first thing that I, before doing this, didn't realize so well is that freezing is such a physical disturbance as well. We are freezing here live under the microscope, and you see now there's like a lot of water movement first, and here's the ice front coming. And it's pushing the organisms, the hyphae, all the minerals around, sorting it quite neatly. And such a incoming ice front can like kill 50% of the organisms very easily. And we have been sometimes chasing some protists, and some didn't survive when these very sharp ice fronts are coming in. And then we have been looking into how organisms interact again here with space. And we found if we do this freezing thawing often, then we have a much higher colonization of the labyrinth spaces than the open spaces. And this is probably because there you have much more refugia left, because the ice front is coming and pushing also all the dissolved salts. They accumulate stronger and lower the freezing point, so you have water pockets in the dead end parts that you can also then very clearly see in these areas. So there's like at -8, there's a lot of areas in the soil that is not frozen yet, where the organisms then happily can continue living. While in the open areas, there's a higher risk of dying there, apparently. And another master thesis was done by Eric, actually out in the field in Kenya. And that was also amazing that we could get off a handheld mini microscope that was doing well enough work to do studies right in the field in Africa. So that was also really nice. He was looking at the effects of biochar on soil microbial communities at the cellular scale now here again. And in order to work with real soil inocula, we have been looking into deep learning image recognition because we, again, of course, want to estimate biomass and cell numbers and so on. So we started with segmenting bacteria, and this is working quite well so far. So we can count bacteria, but not only count, but also then at the same time get their spatial distribution, their size, and morphology. And that is something which is probably a very nice complement to a lot of DNA studies where you don't get all of these informations. So doing both at the same time is something that we're planning to do in our future experiments now. So we can measure the organisms and the biodiversity, and we also want to study the biodiversity much more. We have been starting with pilots, and we're gonna do this summer a lot of field work, looking at a lot of different land use types. And this was the pilot where we were taking some samples from arable field and the adjacent cow meadow, and the communities look like this. Can you guess what was the cow meadow and what was the plowed field? That was in the winter though, but probably you can guess, or so that was quite striking to see that difference. There are organisms, but there's so way less. So hyphal abundance in the meadow compared to the plowed field, like 100 times or 50 times more. Protist abundance, similar, and then also the protist richness, also very, very strong difference. And that then gets us, of course, also to a lot of thoughts, like what can we do to increase soil microbial diversity, soil health? I've been just discussing with Jed as well, like, should we and can we rewild soils? Is that something like, I'm not at all an expert in this field at all, but that's maybe something very interesting to discuss with you here today. I mean, very important things, of course, preserve the structure, like don't destroy the structure and do as much as you can to rebuild soil structure, because that's the home of this microbial diversity. Also vegetate as much as you can. And like sometimes the municipality was asking, like, what can we do in Lund municipality to increase our carbon storage and so on? There's a lot of things that they can do, but like a very simple thing is like, don't mow the grass in the park so much. That's a very easy thing, because like you probably noticed the length of the leaves is very strongly correlated to the root development as well. And of course, all the carbon also then going to the rest of the soil community. Feeding the soil by vegetating, but also like by adding organic matter if it's needed, if you take away organic matter in agriculture, for example. And of course, that's super important to have the healthy ecosystems because they will also be the base for our own health. In the end, you know probably the One Health concept. But in agriculture, it's probably difficult to get a really amazingly healthy soil ecosystem, because that's what we want to have in the end, like something like this, that is full of hyphae and full of different organisms. There's a little ciliate, there's like hundreds of bacteria just in this image here. We need to have a strong, good plant biodiversity and perennials. So there are big challenges for agriculture to be sustainable for their soil ecosystems. And I think I've been realizing more and more the last years that I'm actually maybe in the wrong business, because I'm a natural scientist, but I think all these big issues, these big frontiers for sustainability, they are in the social sciences, because it all comes down to what we decide to do and not to do and so on. And that's why I think it's very important for me personally now also to spread the word of how amazing and how exciting and how special soils are, like all these creatures that there are that we don't know anything about, that we have hardly studied, and like no layperson knows what's going on. under our field. And kids love Pokemon. Yes, that's great. But we can maybe make more realistic Pokemons for them as well, so that they learn something usable at the same time as well. And we just recently started a little YouTube channel where we compile a lot of the videos that we take, and we explain what's happening. And hopefully very soon there will be more. There's what so far is one video up, but more to come, hopefully. And with this, I want to conclude and thank you so much for listening.
So I just wanted to say thank you so much for such an incredible talk. And every time I see those videos, it just blows me away completely for the work you've been doing and the work you're furthering. I'll take this opportunity now to ask some questions, or if there are any questions in the room for Edith. And we'll also probably take some from online as well. Sorry.
Well, I told myself I wouldn't butt in because I'm no longer active in the field, but I'm just blown away, of course. Do you think it's possible that, you know, we have a gut microbiome that everybody's begun to be very aware of, and we're eating flax seeds and blueberries and so on? Do you think there's a soil microbiome that has evolved to create the right sort of spaces and to build its own architecture?
I'm pretty sure that there's super much feedback going on all the time, yeah. I mean, they have been evolving, the microbes have been evolving in different soil parent materials and adapting and creating their own world that they are home in. They have been building their home and they have been evolving in this, I'm pretty sure. And I also like very much the comparison. We're getting more and more aware of all the beneficial and important microbes that inhabit us. And I sometimes say, like, I don't want to have any strange dietary tips or whatever. I think I just need to think I need to feed my microbes when I eat or so. And we have to think the same with our soil. Like, we need to feed our soil because it's a living tissue kind of as well.
Yeah, am I passing you this so that people online can hear?
Yeah, thank you very much. Does it work?
It's
for online. It's for online audience. All right. Sorry. I was wondering what happens if you take one of these tilled field and turn it back into a co-pasture? How do you have any idea how long it takes for the diversity to come back, and also would it make sense to have soil transplants as you would do with the gut microbiota to inseminate back and the lost diversity?
Yeah, absolutely. I think that's a good way to speed up a regeneration of a degraded soil is to have some soil from nearby. We have also been just discussing this an hour ago. The possibility to buy inocula, for example, for mycorrhizal species to put into a foreign environment is probably much less efficient than if you take just an inoculum from nearby of a healthy soil and get a community inoculum. The exact timing of how long it takes for community to reestablish, I don't know. It depends, of course, very much on how degraded the system was, like how degraded the habitat is as well. That's also like a long time ago synchrotron story. My very first time I wanted to look at organic iron in that in soils. Iron is bridging between minerals and organic matter a lot. It's very important for building up structure. I was trying out some techniques, and I didn't get any differences in my experiments with the technique I was trying to measure. And I thought, okay, now I'm gonna measure something that was really extreme, where I last year added 20% volume organic matter into a mesh bag that had been in the soil. There was a Tunisian soil, also very degraded, half desert soil. So that must be like really strongly affected because it got so much organic matter. I still couldn't see this on the molecular level because it takes so much more time for a soil to build up that structure, to have the degradation of organic matter turned into dissolved organic matter and into exudates and stuff that can create those structure and those also organic matter mineral interactions. So there wasn't at all enough with 20% organic matter for a year in a Tunisian soil to make any difference there. So building up from a very degraded habitat can take quite a long time.
Thank you. That was absolutely mind-blowing. Was just fascinated by seeing that, completely blown away. I guess a two-part question. The first one is more sort of social. Is like when you're looking at those videos, like as scientists, you get in trouble for like anthropomorphizing fungi and things like that. But I find it very hard not to sort of look and see like traits, foraging traits as sort of personality and stuff. So that sort of first question is what are your thoughts on that? How do you deal with it? But the second one more actually looking at those traits. So you have that sort of very direct zombie forager and the snake-like one. Have you had any conjectures of sort of the types of soil environments where one might flourish versus the other, and whether there's any way of relating that to the sort of field environments?
Yeah, the zombie fungus, for example, they are all litter decomposers that we have been comparing there in that first study. To your very first question, we don't anthropomorphize them in the scientific papers. We only for a talk. The zombie one goes for much larger chunks of litter, so more like woody parts of small root pieces and so on. So it needs to be much more like protective of the resources that it finds. It has to invest a lot of enzymes, so it's important for it to have a much denser mycelium than some of the others. There's a dung decomposer, for example, that is a really opportunist that has to deal with longer distances to their next research patch and so on, and is therefore more prone for foraging far instead of dense. Even if there's no nutrient reward to any of them in those experiments. Yeah.
Thank you for a truly beautiful talk. So I work on antibiotic resistance, and so I'm fascinated by the ability of some fungi and soil bacteria to make natural product antibiotics. And I was really intrigued by all the different sort of space filling strategies that the fungi have that you show. But the bacteria you studied mainly look like they're sort of planktonic and just swarming around. But there are some filamentous bacteria in soil, like Streptomyces. So is there any prospect of investigating how they behave? I guess the structures have to be much smaller because they're just smaller, but yeah, I'd like to know about that.
Yeah, I also definitely would like to work with filamentous bacteria. We haven't done it yet. It would be possible to construct smaller spaces that even would challenge them. And I mean, like, there's always this thing, like, we want to challenge them, but it's also for microscopy. If you have spaces that are much larger than the organisms, then things overlay a lot, which makes it more difficult to image them. So but it would be technically definitely possible. I've been thinking in, like, now we are especially constraining size exclusion in our systems by the ceiling. It's only 10 microns high. So get quite large organisms, but then they still need to squeeze in the Z direction. And it's possible to make like a stairwise lowering of the ceiling as well at some point. So that would be very nice.
Hi, thank you. Thank you for the talk. So I'm curious, I work a lot with tropical forest, like across the 4 tropical continents. And I was wondering, in one of your slides, you put kind of like the higher number of perhaps of taxa you have for bacteria and fungi at the same time kind of increases, if I understood properly, the composition of organic matter. Well, I'm not sure.
Biomass. There was only one species.
Okay.
Yeah.
So my question would be, what kind of relationship would you expect, like between fungi and bacteria for the composition of biomass or organic material across the tropics? in these kind of pristine ecosystems in which, for example, only climate change, there is no anthropogenic direct effect? And the second is, to what extent would you be able to, if we have a lot of soils from many different tropical forests, would you be interested actually using some of it for your analysis as well that you have been showing?
Absolutely. The interactions between fungi and bacteria are very different depending on the species identity. They often are competitive. In our experiments also usually, like as soon as we put both in, both grow on a bit lower level because they share the same resources. When we have been looking at soil inocula, where we have a large variety, we see often also mutualistic interactions, or at least bacteria that are very happy to follow fungi, both because they probably get a little bit of free meal there, but also because of the hydraulic conductivity that the fungi drag water films with them, so they can only move across air pockets via them. But then we have been showing in the study that the amount of fungal hyphae in a pore space is very strongly predictive for the amount of bacteria we find in there. Because of this, they only can move and disperse over air pockets when there is fungi making water bridges. But not in all cases. There was quite some variation because I think there are quite some antibiotic producers. Like in some areas, there's hyphae, but it's completely bacteria-free as well. So there's a lot of different combinations possible.
I think we're going to take some questions from online, so I'm going to pass over to Stephen.
Just one online question from Sarah. She's from a climate action group that's got a film called 6 Inches of Soil. But her question, I think you mentioned, just relates about the YouTube video that you're doing, and how can you make this kind of research and presentation more accessible to schools and children? And is this where some of the changes that we need to see are going to be about kind of inspiring the next generation to think more about soil? And so I don't know if you could maybe mention what the YouTube channel is and sort of what reactions you've had from this kind of work with younger school children and so on.
Yeah, this is still very new, so not super much reactions yet. I think like we have like 13 pre-numerations yet, so not very, very early. But I totally agree with Sarah that it's super important to get children closer to soil biology. And we just have been ordering more of these field microscopes as well, because the one that was with the master student in Kenya, we left in Kenya for them, so that we have something to live be able to look at soil communities also then in the classroom. And that's something I want to do a little bit on the site in the future as well.
Brilliant. So if we have no more questions, I just want to say thank you so much again for such a wonderful presentation. And yeah, and there will be a drinks reception in this room afterwards. So please stick around and mill around and have lovely chats. And yeah, thank you very much again. And we'll see everyone at the next one.
Thank you.